Let me begin. Hello, everybody. My name is Most Worshipful Bill Sardone, Grand Master of Masons in the state of New York. Thank you for joining us this evening. It's a pleasure to have all of you with us. We've all been hearing too many things about the words COVID, pandemic, stay at home, social distancing, but there's some names that I never get tired of listening to, and one of those names is Dr. Maria Contreras, and she is joining us tonight with members of her staff and scientists who work with us at the Masonic Medical Research Institute. This institute has a long history of not only doing things here in New York, but globally, and Dr. Contreras and her team have put this on the map. Tonight is coming in here thanks to a number of people. Varun has put us on some level ground at the institute itself. Brother Anthony Cucci, Kathy Bartels, Right Worshipful Ed Bartels, obviously the staff and admin that work at the Research Institute, and the board of directors who work there, who are volunteers and who are all Masons, to see that we're doing the right thing and working together with their skills to put this in the right direction. But most of all is the tens of thousands of Masons who are in the state of New York who continually donate and stand by this fraternity, the Masonic Care Community, and the Masonic Medical Research Institute that makes everything work. During this period of time of shutdown, some people say that we are losing ground. Here in Masonry, we're actually gaining ground. We may be apart, but we're actually coming closer together, which is the bond of brotherhood that this fraternity propagates. So at this time, I'd like to also say that we have individuals coming to us, not just from New York State, not just from here in the United States, but I'm seeing here we have people from Bulgaria. I see Nadia's here from Bulgaria. She's a good supporter of everything we do. Baja California, Canada, Spain, and we have some people here as far away as this unknown city called the Bronx. I didn't think we had anybody from the Bronx here, but there they are. They have come on with us. We have a Q&A opportunity and portion down on the bottom of the screen. We'd appreciate it if you would use that to put your Q&As in. There will be an opportunity to answer some of these questions. I know there's a chat box as well, but try and keep your questions into the Q&A. At this time, the Executive Director, Dr. Maria Contreras, I turn this over to you. If I were to be here tonight and speak to the titles and accomplishments that you have, that would eat up the entire webinar time. So I appreciate your being with us and with your team. Dr. Contreras.
Thank you so much, Grandmaster. It's truly an honor to be here tonight with all of you, and thank you to the Grandmaster for the kind invitation to give you an update about some of the wonderful things that we've been doing here at the Masonic Medical Research Institute. And with me, I brought several individuals who you're going to hear a little bit from in a minute, talking about some of the research that's going on here at MMRI that's leading the edge to COVID research worldwide. So first of all, I'd like to say, for those of you who may not know who we are, let me just start the slide presentation here. Just one second. Okay. For those of you who may not know who we are, we're a small institute. Sorry. Trying to get back here. There we go. We're a small institute that was founded in 1958 who has grown significantly over the last few years. This is a photograph of us a couple of years ago now from 2018, and you can see that we are already expanded. We started at about 17 people here in 2018. We're close to 50 people now doing really state-of-the-art science and research that I'm going to tell you about in just a few minutes. And we really span the scope of work from anywhere from cardiovascular research to autoimmune disease to genetic diseases and new technologies. So what have we done since 2018? We've actually rebuilt a lot of this building, which was basically built in 1958 and hadn't been touched until 2018. The renovations have really brought us into the 21st century with state-of-the-art equipment, state-of-the-art space, to continue to grow and expand our research projects. We've been publishing in top-tier manuscripts that are worldwide and world-renowned. We've recruited amazing scientists from the top research institutions in the country, and we've been bridging the academic network and collaboration with MMRI and other institutions and pharmaceutical companies around the country. And most importantly, for the continuation of our research, we have actually increased all of our grant revenue that comes into the research enterprise to allow us to continue our research efforts going forward. So what is our mission and vision? We want to improve the health and quality of life for all humankind. And this means that we need to conduct high quality biomedical and clinical research. And part of that research is training and teaching basic scientists, clinicians, and students who can perpetuate and extend this fight against disease worldwide. We understand the molecular biology and the molecular techniques that are required to get us to the next step, and the hope is that we will develop medical cures and treatments for tomorrow. Our research strengths really span four major areas: genetics, molecular biology, metabolism, and technology. In terms of genetics, we're really advancing the field with respect to personalized medicine, working on everything from cardiac disease to autism and autoimmunity. In terms of our molecular biology, we use this as a basic tool to understand how you develop a disease. And we focus on pediatric genetic disorders to inflammation and adult end-stage heart failure. In terms of metabolomics, we're working very hard to understand the role of obesity and aging and heart disease. And with respect to technology, we're advancing the field with nanotechnology, nanoparticle technology, gene therapy delivery mechanisms, as well as inducible pluripotent stem cells in order to enable us to answer how we get disease, how we can treat it, and eventually find a cure. The basic pipeline is as follows, which is, I think, really important to understand how we actually get to a point of getting a drug out in the market for treating diseases. We start at the basic science level, which is what we do here at the Masonic Medical Research Institute, which is primarily focused on identifying the mechanisms that cause disease and then finding the targets to treat them with various therapeutics. That then moves into a in vitro translation stage. And what that means is that we do cell culture work that allows us to validate those targets and use human tissues and samples as biomarkers and readouts for those studies. That then moves to the third phase, which is drug screening, to identify, generate, and validate the drugs and target molecules so that we can get the validation we need to move to the final stage, which requires collaboration with multiple sites as well as pharmaceutical companies to test in larger animal models and to do clinical trials in humans so that we can get a drug into the marketplace. So how does this all relate to coronavirus? And that's what we're really going to focus on this evening in our discussions. But I thought I would introduce the disease to all of you in case you haven't heard enough about it in the media in the last couple of months. But basically, severe acute respiratory syndrome coronavirus 2 2019, or COVID-19, is a new virus, and it was first identified as pneumonia in Wuhan, China in late December 2019. We think it probably started in animals, predominantly the bat background, and now has jumped into humans, and so it's spreading between people at a rapid rate. So, and because this virus is new, we're learning more about it every day, and everything is changing from a day-to-day basis, as you can see from what's being reported from the scientific literature as well as the media. How is it spread? As you've come to realize, it's being spread as people get infected from one another. It spreads the same exact way as colds and flus, with a little bit more intensity, and it's through droplets that we sneeze or rub our noses or touch our faces, with which allows for that rapid transmission. So the goal is to try to avoid touching your face, touching your eyes, and touching your mouth. So this is why I think wearing masks is so very important. Not only does it prevent you from transmitting the particles from your mouth to other individuals, but it also prevents you from actually touching the surface of your face by wearing them. These also can be transmitted from people who are displaying any symptoms, which is why this disease has become so rampant. And that's a really important touch point that I think we're going to focus on in a little bit. What do the symptoms look like? You have fever, that's one of the major symptoms, a cough, sore throat, fatigue, shortness of breath, and really specific to COVID-19 is the sudden loss of smell and taste, headaches, muscle aches, and sometimes diarrhea. The symptoms can range anywhere from 1 day to 14 days before their onset, but predominantly it's about 3 to 7 days after exposure where you start to see some symptoms. And as I mentioned earlier, some people show no symptoms at all or have very mild illness, but that doesn't mean that this can't transmit to somebody else who may actually react in a much more negative way, which is why we have to work very hard to protect each other as this pandemic makes its way through the rest of the country. So how do we do that? I mentioned hygiene. That's the one and most important way that you can prevent transmission of this disease. Wash your hands frequently, as we've talked about previously. Make sure you use an alcohol-based hand sanitizer when you can't get to soap and water being the best thing that you can use to get rid of this virus. Cover your coughs and sneezes with a mask. Use tissues and your upper sleeve. Don't touch your face if possible, and make sure you throw away everything into a bin and wash your hands. Don't share food and drinks, and that includes your cell phones. Make sure you clean them on a regular basis. If you have any symptoms, there are guidelines now that you can follow through the CDC, federal and state guidelines that will tell you how to proceed with getting tested. In terms of diagnosis and treatment, because there are so many other illnesses that are similar to COVID, it's very hard to always know that this is specific to COVID and you don't have allergies or another kind of cold. So swab testing through nose and throat swabs and a blood test are the best way to determine whether you have active disease or if you have had the disease in the past. And we're getting to learn a little bit more about what that means in terms of antibody testing with the blood test and how long those antibodies are protective once you've had this disease or not. Currently, there is no specific treatment for COVID-19. The best situation is to not get infected. If you do get infected, there are now better ways that we are understanding for how to treat this disease. Mild symptoms can be treated with medicine to lower your fever and relieve the pain. And for anyone who's suffering through severe symptoms, they should go see a doctor or go to the emergency room right away. And as many of you are aware, a lot of this is now currently being propagated through vaccine treatments. There is a vaccine currently in clinical trials here in upstate New York. I read in the newspaper just started this week in Binghamton with a potential phase 1 clinical trial through Moderna, which is the first company here in the States to do this. So with that, I'm going to stop and with that introduction and start to introduce my panelists who are going to talk a little bit more about their research and what it means to translate their work into COVID-19 related research. So our first panelist is Dr. Chase Kessinger, shown here. He received his PhD from the University of Texas Southwestern Medical Center. He did his postdoctoral training with Dr. Farouk Jafar at Mass General Hospital in Boston and Harvard Medical School. And in 2015, and as a testament to his achievements, he was promoted to instructor of medicine at MGH and Harvard Medical School. We were really fortunate to recruit him to MMRI in 2018. We think he's a potentially high impact faculty member who's going to be doing research on imaging and histology. He is actually running our core facilities for imaging, histology, and surgery currently at the Institute. And his area of research is focused on vascular cancer biology, deep venous thrombosis, nanomedicine, and imaging modalities that can assist in generating targeted strategies and device treatments for cardiovascular disease. Through his hard work and efforts and dedication to the field of deep vein thrombosis in particular, Chase has expanded his research to focus more specifically now on pulmonary embolism as it relates to COVID-19. So with that, take it away, Chase.
Thank you, Maria, for the introduction. I wonder if my audio is good because my headset just crapped out.
We can hear you. Yeah.
Okay.
Good.
So, like Maria said, today I'll be talking about COVID-19 and blood clots. So, let me jump in. I'd like to start with basically the idea that the body is always in some kind of balancing act. And this has positive and negative feedback loops that can help make changes over short and long periods of time. Now, let's say the body is challenged by a virus, that being COVID-19. So what does it do? It initiates basically these protective systems to help the body deal with the infection. And one or two of these protective systems involve inflammation as well as activating your immune system. The problem is if it's overactivated, this can lead to some damage to other organs, especially the lungs in this case, and sometimes, as we'll get to know in the talk, the heart as well. So I'd just like to say we always want to position ourselves to be healthy. That is, to put ourselves in the correct place so that we can fight these things. And during this time of COVID, it's important to always stay active, either that being a lockdown inside of your house or also, taking short walks outside as well, as long as everybody is safe. And also remember to keep up with your medical therapies, going to the doctor and things like that. So what I'll be talking about is basically my focus for my research, which is blood clots or venous thromboembolism or VTE. Basically what this is the venous stands for the vein, thrombo stands for blood clots in the vein, and then embolism is when those blood clots travel from the legs, as in DVT, into the enlarge in the lung, which is now termed pulmonary embolism. So VTE equals DVT and PE, so thrombosis in the legs and also those blood clots in the lung. So why is that important to just be a research area in general? Well, this affects about 10 million people across the world each year, and it's estimated that VTE related events kill more than AIDS, breast cancer, prostate cancer, motor vehicle crashes combined. There's also a huge socioeconomic burden with VTE in that we use billions of dollars to treat and diagnose this in the hospitals, but there's also a major loss in total, that being quality of life loss, that being you're staying at home, you can't walk, you can't go to work, and that actually totals closer to 50 billion per year. So what do we need? We need new tools in the clinics, we need new strategies, new therapies, new ways for doctors to diagnose these diseases, as well as really beneficial ways to treat it. So that brings us to how COVID is seen and how do blood clots relate to what we're seeing in COVID. So as Maria mentioned, symptoms range from mild to severe. And these patients that are being admitted to the hospitals are showing high markers of inflammation as well as activated coagulation. So their body is actively creating blood clots and trying to lyse them as well. What we're seeing is that 30% of these patients that are admitted to the ICU, so the severest of us that get the illness, are actually coming down with about 30% of them are coming down with a VTE event or a blood clot, that being either in the lungs or in the deep veins. So from this early knowledge coming out of China, we've actually started to give patients that come in with COVID and admitted to the hospitals, they're already started on anticoagulants, or as we know, blood thinners, to help fight this onslaught of blood clots. So what do blood clots mean for this infection? Well, in the healthy individual that we have on the right, basically at the terminal end of when you breathe in with your air, you have these alveolar sacs, and basically what they're meant to do is exchange oxygen with your blood. Now, when you start to have moderate illness, these sacs, due to inflammation and the infiltrates of inflammatory cells, basically start to fill up with fluid to try to protect themselves, as well as due to high inflammatory burden. So as you can imagine, if you fill up an air sac with water, you can no longer change the oxygen. And that is why you're starting to see why people are having trouble breathing. It's interesting to see here is a patient that actually succumbed to the disease, but these black dots are all the inflammatory cells. And you can appreciate in the healthy individual over here on the left that those black dots are absent. And these are all inflammation based. Now, in the severest of cases, what happens is that whole sac or that whole region of the lung basically is filled with fluid, and you start to get microthrombi or thrombus that forms in these areas here in the blood vessels themselves. That can be appreciated here is that there's a healthy lung exchange capillaries here, and then as the microthrombi form, the blood vessels are actually trying to reform to try to get around these blood clots, and you can see the basically disastrous results here in that the blood vessels don't know what to do. They're trying as hard as possible to regenerate, but don't really make it. So what does the clinician see? This is kind of important to us at the MMRI because we also have these kinds of imaging modalities to image small animals, basically to mark their progress in these types of lung injuries. And what we see is over day 1 through day 11, you see this growing type of opaque whiteness in the lung. And this is actually a patient with COVID. And these parts of the lung are now unusable for air exchange. So this person's having difficulty breathing, their oxygen level percent O2 is going down. The good news is this is a younger adult where she started to basically improve a couple days later, and you can see that this whiteness went away and started to turn black. So black is air in this. So the question is does this heal completely? Can we get rid of this completely? No one knows right now, but we can glean some data from the first SARS back in 2003, which tells us that most of it does heal, but there is a percentage, about 20% or less of the patients that actually have impaired lung function out to 15 years. So how do we improve on this? What therapies could help that? So that's kind of my area. We're trying to evaluate therapies using small animal models to look at this. So one of the things that we published on recently, or a couple years back now, is that statins. So statins are your cholesterol lowering drugs. I'm sure we're all very aware of these. They actually have other interesting side effects, benefits, to where they help to reduce the inflammation of the body. And in this case, when talking about blood clots, they actually help reduce the amount of blood clot and the damaging side effects from these blood clots. So could that be a helpful thing for people with COVID? We're trying to see. Also, what we've been able to do is show that the age of the blood clot is important. So a physician needs to know when the blood clot was formed and how to treat it. That it definitely would inform him on what further steps to take. As I said, statins do lessen the inflammatory response, especially in the deep veins, is what we studied. And currently there are small studies here and around the world that are starting to put patients onto statin therapies as they're admitted to the hospital, see if there's a benefit. So we'll wait and see. So what does this look like? Our research, how do we get it done? Basically, our science is multidisciplinary. And as Maria pointed out earlier, there's multiple different investigators with strengths in different fields, these being cell biology, imaging, inflammation, and vascular biology. All these things need to integrate to make these kinds of translational, basically translational findings that we can directly apply to clinical research. So what we do is, or what I do is basically use these imaging strategies, as you can see here, and basically create these diseases in these animal models, and we can track where and how hard or how much the body is affected by these infections or challenges with blood clots. Interestingly, if you look here, these little dots here that are colored in rainbow are actually blood clots in the lungs that we can actually evaluate in the animal, follow the progress of them, and quantitate how well they're either resolving or staying in the animal due to therapies. So with that, due to the inflammatory response and the virus's characteristic damage to the lungs, I just want to remind everybody that it can also damage other organs, which Sathya is about to talk about, and that mainly is the cardiovascular system. It is important to note that when you do damage the lungs, you also are basically causing an acute stress on the heart in general. So there's that to keep in mind as well. So with that, I think we'll hand off to Sathya.
All right, thank you so much, Chase. I'm delighted to introduce our next panelist, Dr. Sathya Nadarthi. Dr. Nadarthi is a talented young investigator who we were fortunate to recently recruit to MMRI. He received his PhD from the National University of Singapore and did his postdoctoral training under the tutelage of Dr. Thomas Hund at the Ohio State University, where his research focused primarily on studying cardiac arrhythmias and heart failure. In recognition of his achievements, Sathya was awarded an American Heart Association postdoctoral fellowship for his training. Upon moving to MMRI earlier this year, Dr. Unadarthi's research interests were expanded to focus on the role of cardiac inflammatory components in development of heart disease. And in particular, he's been working to understand the mechanisms leading to hyperinflammation in the heart in response to COVID-19. Sathya, take it away.
Well, thank you. Can you all hear me okay?
Yes.
All right.
Yes.
Thank you, Grandmaster, for giving us this opportunity. I really appreciate it. And also, Maria, thanks for the generous introduction. So like Maria mentioned, my interest, if I can control my mouse. Uh-oh, okay, give me 1 second here. Let's see this slideshow. Okay, so my interest is in understanding the relationship between COVID and cardiovascular diseases, also called CVD. You know, if you look at the data we had in the past 6 months, ever since the pandemic started, we see that CVDs or heart disease is one of the most common comorbidity associated with COVID-19. Now, what does that mean? What it means is if you take a group of COVID-19 positive people and ask the question, what other diseases do these people have the most? Then CVDs or heart diseases come up at the top of the list. So a lot of COVID positive people also tend to have a heart disease. Now, in addition to that, if you look at the mortality rate or the fatality rate, people who have both COVID-19 and heart disease have a higher fatality rate than other comorbidities, like let's say people who have COVID and Alzheimer's or COVID and kidney disease. You know, what is more worrisome that people have started to find out recently is that COVID-19 actually can trigger a heart disease in patients who have no prior history of heart disease. And we are trying to understand how is that happening. So hyperinflammation or excessive inflammation that Chase mentioned before is one of the main reasons or main mechanisms through which COVID-19 damages the body. You know, if you look at it, the damage caused directly by the virus, the SARS virus, is actually lesser than the damage caused by our body's response to the virus. When the body senses the virus, it turns on the inflammation to a really maximum state, and that inflammation is what causes severe damage, almost leading to death in multiple cases. So if we want to control the harmful effects of COVID-19, we need to learn how to control the inflammation. And my area of research was in looking at inflammation, specifically in heart. So I was interested in two specific proteins, and I'm currently working on them. One is called TWEAK, which stands for TNF, like weak inducer of apoptosis. And it binds to a protein which is expressed on the surface of cells called FN14. You know, you can look at it as a key and a lock. FN14 is the lock, and TWEAK is the key that goes and binds to the lock. And once the key binds to the lock, it opens up the door to inflammation, and inflammation goes up in multiple tissues. So I previously demonstrated that if we can somehow block this lock and prevent the key from binding to the lock, we can reduce inflammation, specifically in heart. For this, we've used a mutant mouse. We used a mouse and we removed this lock in the mouse by using genetic engineering methods. And before I show you that data and tell you how is it related to COVID, I would like to give you a quick Cliff Notes version of how inflammation damages the heart. You know, once the heart has an injury, before the heart goes into heart failure, there are these sequence and series of events that happen inside the heart. You know, it's almost like dominoes. One event leads to the other event, and the second event leads to the third event. And when they all happen in that sequence, eventually the heart undergoes failure. So if we want to control the damage caused by inflammation to the heart, we have to stop one of these events or control those events so that we can prevent this progression. Now, let's take a look at what happens when we block one such an event. And we did an experiment. Grandmaster Sardone, would you be okay with going through this data with me? We can make it interactive.
Have at it. Have a good time.
All right. So what we did in this experiment is we took a normal mouse, and then this is called the wild type or control. And then we took another mouse wherein we deleted the lock, which is the FN14. We call it the FN14 knockout mouse. And we subjected both these mice to a heart injury called TAC. And after a few days, we looked at their hearts. And whenever you look at macrophages, when these macrophage cells enter into the heart, and when we stain them, we look at them as brown dots in the heart. So Grandmaster, between these two animals, where do you see more brown dots?
Well,
Is
if
it
you're
the top?
colorblind, if you're colorblind, you can't be a scientist.
So what we see is, you can see most of these arrows, greater number of arrows on the top panel. Is that correct?
That's correct. Correct.
So we see more brown dots entering into the normal mouse. But when we delete that lock, the FN14, when we remove that from the mouse, we don't see those brown dots or these cells enter into the heart even after injury. So what this means is, by removing this protein, we were able to stop the process or control the process, tone it down at step 2. Now, like I said, this is a sequence of events. So now let's look at what happens in almost step 4. So the blue color you see is an indicator of an activation of step 4, which is just a step before heart failure. Now, again, let's compare these two images. You can see that the top panel has more blue color than the bottom panel, suggesting that the normal mouse was entered into is going towards heart failure, whereas the mouse wherein we deleted the protein is not going into heart failure. It kind of paused at this stage. Now we further analyze the function of the heart using sound waves. We call it a 2D echocardiography. What we do is we put sound waves onto the heart and we look at the heart function. If the heart is contracting well, we see waves like you see here. But if the heart is not contracting well, they almost looks like stationary or flat lines, and that's what we see here. So this experiment suggested that when we knock out this protein, we can control the harmful effects of inflammation on heart. Now, how does this relate to COVID-19? So this is the heart section of a 69-year-old person who died due to COVID-19 in Italy. And they took his heart and they were staining for the immune cells. Similar to what we see on the top panel, we do see these brown dots in the heart section, suggesting that macrophages, the second stage, got activated in this person. They also found virus particles in the heart tissue in this patient, suggesting that in few individuals, it is possible that the virus can actually directly reach the heart. And they were, in this study, which just came out 2 weeks ago, people looked at what happens when almost 2 months after COVID recovery. They were looking at a group of 100 patients and they were trying to see what happens 2 months after they recover from COVID using MRI, cardiac MRI. So they found that even after 2 months after recovery, 78% had abnormal cardiac MRI findings, suggesting that there was either increased fibrosis or increased edema in the hearts. Also, 60% had an ongoing inflammation reaction still in the heart, even after 2 months of recovering from COVID. And these patients also had lower left ventricular and right ventricular ejection fraction, which is a measure of how well the heart is pumping the blood. So using a mouse model, we already showed that if we can remove this protein, FN14, we could prevent the heart from going into advanced stage failure. So my research at MMRI is to say, can we replicate this with COVID-19 patients? So we are currently doing experiments with cells wherein I'm using a small chemical molecule that can block this FN protein on cells. And I'm asking the question, if I block it, can I prevent COVID-induced inflammation and COVID-induced heart failure? So that is what I'm working on right now. And I thank you for giving me this opportunity.
Wow.
Right. Thank you so much, Sathya. And if Chase can advance our slides just a little bit more. I'll start by introducing our final panelist, which is Dr. Nate Tucker. Dr. Tucker is actually our newest faculty recruit, came also in March of this year, also from the Massachusetts General Hospital and Harvard Medical School, as well as the Broad Institute in Boston. He was originally from the upstate New York area and moved after his bachelor's degree at Syracuse University to Washington State University for his PhD, and then did his postdoctoral training under the guidance of Dr. Patrick Eleanor, who is a world-renowned cardiac electrophysiologist and geneticist. Nate, too, was promoted to instructor at the Mass General Hospital in 2016, which is also a testament to his potential as an independent scientist and researcher. He joined MMRI this year and has already taken the bull by the horns, so to speak, and is running our COVID-19 testing facility in partnership with the Mohawk Valley Health System here in Utica, New York. And he's also helping us to he's assisting the MMRI in reopening the safety and health of our community at large. In addition, Nate's research is focused on genetic mechanisms underlying the risk of atrial fibrillation and other cardiovascular diseases, and he's been actively working on understanding the genetic etiology of COVID-19 in the heart on a single cell basis, and where he's already published at least 2 to 3 papers that are coming out actually this week on, or this next couple of weeks in Circulation, which is an amazing journal in the field. So take it away, Nate.
Thanks for the introduction, Maria. Thank you, Grandmaster Sardone, for the opportunity to present, and thank you to all the people viewing. I'm going to split this into 2 parts. One will be a bit about our testing program here at the Institute, and then the other a small tidbit about the research that I think is going to answer part of the one question I already saw from Mr. Alden Bush about distribution. A very good question about distribution of the receptor. So I'll start with epidemiology and clinical testing that we're doing here at the Institute. So I strongly suggest if people are interested in what's happening, both from a testing standpoint, a case standpoint, you know, these other statistics, the most wonderful resource you can find to view these is a website called covidtracking.com. It's actually consists of a volunteer force that aggregates these data. So I think speaks to a good spirit of the people that are involved in this particular project to give the information to everybody in a digestible way. So just to give a little overview of where things are at in the country. So the left-hand graph is going to display the amount of tests that we're doing as a country. So a pretty linear increase through time. I don't think we're anywhere near what we're going to need to be in order to fully reopen at some point. If you look at the second graph here, that's the number of cases. So this first hump that's kind of hidden would be the one that was present here in the Northeast. The lull, after which most things reopened, and then the more recent hump being the outbreaks in the South and Southwest. You can see similar trends with hospitalization and then daily deaths as well. You know, the serious situation we had here in New York driving a lot of this early death, and now a small uptick corresponding to the recent case increase as well. So along with this website, you know, you can dig through particular regions. So this is the trend for the Northeast, thinking New York, Massachusetts, Connecticut, New Jersey, et cetera. So our testing capacity increasing as well. Number of cases are low and are staying low and reducing the number of individuals hospitalized or dying from this condition. The case is not the same if you were going to look in the South. So they missed that first bump that we got and are now experiencing that surge that hopefully we dealt with and completed a few months ago here in New York. A few things I wanted to talk a bit about New York State, but since Grandmaster Sardone mentioned that we have folks from overseas, I thought I'd give a little context about where we're talking about. So a picture of the US. The blue dot is actually our institute in the center of upstate New York. We can look at the number of cases that were throughout the state as a whole. This is not adjusted for population, and they capped it at 20,000. So if they made more scale down here in New York City, it would obviously be much, much darker. Upstates made it out a lot easier, actually. I mean, as a percentage of population, perhaps due to, you know, being a mostly rural population or some of the safeguards that were put in place after the extreme outbreak down in the city. we've been pretty fortunate up here. There's some statistics for the state. We're a very small percentage of the testing that's occurred, although that's increasing as we go on. About 60,000 a day we're up to, and that's the total number of positives. Again, the vast majority of them downstate. If we look at trends for people tested and the number of positives, we've just started to level off on the number of tests. I'm happy to talk a lot about the testing infrastructure if people are interested. And the number of positive cases we're seeing in our hands and in the data of the state seems to be leveling off. We're at about 2% to 5% positivity for our tests, and we haven't seen a lot of fluctuation from that in the last few weeks. So hopefully that's a positive sign for New York State. The breakdown of mortality or fatalities to this is certainly a sad statistic to look at, but the risk of mortality in folks who are older is definitely drives the most of the death that we've seen. In young people, the risk of death is relatively low, although we're learning a lot about the other side effects that may come from infection. So when we talk about testing, Maria alluded to it earlier, there's two major types of tests. The top red one here is going to be what we call a molecular test. So what that does is you're going into your walk-up clinic or one of the drive-throughs, they're sticking the swab up your nose, putting that into a particular liquid, and it gets sent to our facility or others. And what we're looking for is if the virus is actually there at this moment. So much like we have a genome that makes up who we are, the virus has one as well that's distinct from us. So we can actually see if it's present within you. Now the other testing that's being done, not as much, honestly. The reliability of antibody testing's pretty poor, and I think it's gone out of favor for monitoring past exposure, but there's still a low level that would say, perhaps you've been exposed to it in the past and would hopefully be more safe against future infection. So we're doing the above. And what we did was really what I'm going to call a community-based solution. So when I got here, I think actually on site in April, we all wanted collectively to figure out what we could do to help. We were still in phase 0 of reopening at that point. So one of the big barriers was the testing capacity for a given region to be able to reopen. So we were scrambling with the state and trying to see what we could do to perform these tests. We had the people, we had the facility, we had the instrumentation, we had the expertise to do all of this testing, and just really needed a partner to make the clinical operation happen. We were fortunate enough that some of the individuals at the state actually suggested partnerships with a local hospital. We reached out to the Mohawk Valley Health System. This is an image of what their building will look like 3 years from now. They're now split across several campuses in the Utica area. And we're able, within the period of 3 weeks, to go from an idea to actually performing our first tests within our facility. So much like, eating local food, I think meets a certain need for all of us. I mean, we're able to take care of the carriers of our community from a testing standpoint here within the Mohawk Valley as well. So we commenced on June 1st. I want to give all the credit to these two individuals over here. So if any of you happen to visit our facility once we're more open, this is Ryan Pfeiffer and Mayuri Desai. They're our two certified lab technicians. I'm really the person who helps make all of this work actually happen. So since the first, we've done a little over 5,600 molecular tests, 400 or so positive. Our turnaround time, if we receive a sample before 1 o'clock in the afternoon from the hospital, that's turned around in the same day. A lot of places are experiencing issues getting tests back within days, and we're able to turn these around, within hours most of the time. And what does that allow us to do? I think 3 important things for the community: helping patients go home. In order to be discharged home or to a nursing home, people need to have negative tests and multiple negative tests. So we're able to facilitate for that people clear the beds in the hospital for those who would need it, and help people go back to the living situation that they would prefer. The hospital was unable to perform surgeries and outpatient procedures without this testing. So part of the requirements to do, what would be a normal procedures was a negative test before they'd let you in the hospital. Now we get these individual swabs sent to us, and we'll turn them around fast so that they can get these necessary procedures done. And then finally, the positive tests, particularly that we get, we partnered with the New York State Department of Health to monitor how the virus is actually moving through the state. I'm happy to expand upon how specifically we do that later, but you can look at particular types of the virus and try to ascertain where it came from, especially when partnered with contact tracing information for anybody who's positive. So that's a bit about our testing program. I want to go into one quick research project. So Sathya in particular talked about the effects of the virus on the heart, and one of my projects addressed that at least indirectly, and we're working on directly in the future. So it's estimated that about a quarter of mortality related to COVID-19 is actually due in large part to what we'd call cardiac involvement. So whether it's heart failure that manifests, it's irrecoverable. So there are a few outstanding questions that I think we were in the midst of a project and were able to pivot really quickly and help provide information for. So the three questions I'll say: number one, how is the heart being affected or infected? Sathya showed that one picture that there is some virus in the heart. That's an outstanding question that we're still trying to address. Number two, are there individuals who are at higher risk for cardiac symptoms? And I'll talk about this from a biology basis rather than looking at what we'd call epidemiology or study of the spread by medical records. And then finally, one really important question was early on, there was concerns about the use of what we call hypertension, antihypertensive medications or blood pressure medications, and how that actually interacts or might change the receptor for this virus and might increase risk for infection. So without going too deep into the specifics of what we do, this is a picture of a post-mortem heart. So this is the large pumping chamber of your heart. It's the left ventricle, sends blood to the extremities of your body. This particular individual had a heart attack, at least one at one point. So that's the scar that would lead to decreased output from that heart. So when we talk about heart, it's important that we understand that the heart is actually a mixture of many different things that need to work together. So we think of it as a muscle, but in order to support that muscle, you need the vasculature system to supply the blood. You need some of those inflammatory cells that Sathya talked about previously to help clear out any damage that might have been produced. So it's a really complex system that works together. And really only in the past couple of years have we been able to pull all of these things apart and look at them in isolation. So if I was going to zoom in on this, vast majority of all this pink you're going to see is the working muscle. But if you were going to zoom in greatly, you can see cells and necessary cells that lie in between and help support this performance function. This organ has to beat 2 to 3 billion times in your lifetime, and it never ever gets a break. So all of these things working well and in concert is very, very important. So how we do to tease these things apart and look at all of them in isolation is actually using a trick here. Hopefully the video works for everybody's stream, but we can actually separate these all out into their individual cells, pass them through these pretty neat microfluidic devices that'll separate each one out into its own little droplet, and then analyze what makes that cell unique. And the end result is a picture that looks like this. So without going into too much what it is, if you're going to see little dots here, that's one cell. So this is a study of about 300,000 cells that we published nearly 2 months ago. So within each one of these points is a couple thousand pieces of data across these couple 100,000 cells. So you're talking the millions and millions of pieces of data. And from these data, we're able to answer those 3 questions that I proposed earlier. So number one, the receptor for the virus, where is it found within the heart, the highest expressions, or the most receptor that would be open for infection, are in these cells called pericytes. And what these pericytes do is we think about our coronary arteries, which are the big vessels in your heart. Those actually form smaller and smaller vessels to supply the tissue as they kind of spread through the heart. And those things that line the smallest microscopic vessels in your heart are the ones at most danger of being infected by the virus. Point two, people with pre-existing conditions. So for one thing we were able to look at is people in heart failure. You know, heart failure has various stages. People can live in it a long time, but we looked at people with heart failure, their tissue, and we're able to see in one particular cell, and probably the one as we think of most critical for the function of the heart. We call them cardiomyocytes. Those are the muscle. They do the work to actually pump the blood. Those particular cells in people with pre-existing conditions have more of this receptor and would be more susceptible to an infection. And then probably most important for a lot of the people viewing now, we looked at the antihypertensive medications. We call them ACE inhibitors, and didn't see anything. So didn't see any difference between people who take them or people who don't. So along the lines of what the cardiac societies have been suggesting, you know, we also suggest that, you know, please don't modify how you're taking these very important medications. All right, so what are we going to do moving forward? There's several, you know, on both of the fronts I talked previously, things that we need to do here at the Institute. We're actively monitoring outbreaks, not only in the Mohawk Valley, but beyond, particularly in partnership with the state. I think we've been able, you know, from logistical and equipment standpoints, to expand the amount of testing that we're able to do on a daily basis, and working out that in the future is hopefully will be helpful not only for our area but elsewhere. Flu season's coming, and it's going to be a challenging issue. You know, anybody who's sick will have to be tested for both of these. So we're working actively with the hospital to make testing for the strains of flu to be part of our regular protocol, so that hopefully business from a health standpoint can continue as usual. And then finally, along with what Satya mentioned earlier, I partnered with a researcher at Columbia University who, unfortunately, was a primary care for a lot of individuals who passed from this virus, and we're able to find cardiac tissue and say, you know, answer the question if the virus itself got into the heart, and if that's some of what underlies the damage and unfortunate death that occurred in these individuals. And part of the reason I think everyone would want, you know, what can we do for this virus? And it's, you know, outside of development of vaccines, you know, new treatments, development of new therapeutics probably won't be in time. So unfortunately, what we need to start preparing for is the next one. Chase mentioned earlier, a lot of what we learned about this virus was actually done in the early aughts on SARS-1, which was mostly contained to East Asia. And you all probably remember a pretty severe outbreak in Toronto, Ontario. You know, we need to make the same effort to understand SARS-CoV-2, that's that current coronavirus, in order that, you know, we're ready to meet, unfortunately, the inevitable SARS-CoV-3. With that, I'll hopefully get back to your question here in a second, Alden, but I'll let Maria finish off the presentation.
Okay, wonderful. Thank you all so very much to the panelists and again to Grandmaster. And I thought I would close our presentation with, I think, an apropos quote from Marie Curie, who basically said that nothing in life is to be feared, it is only to be understood. And now is the time to understand more so that we may fear less. I think that this resonates, I think, with all of us with respect to the coronavirus and where we are today. And the information that our scientists and those around the country and around the world working so fearlessly on at the moment will help us get to a point where we understand it and can fight it and can cure it. So with that, I think we're happy to take questions and address any concerns that any of you might have.
There are some questions that came into the Q&A. If you'd like to just take them, I know that Nate and Alden are now going to be pen pals.
Yeah.
But the,
So
but
why don't we start with Nate? Why don't you take the first question from Alden Bush on ACE2 receptor sites?
Sure. So the question for everyone, if you're not able to read it, is has the MMRI done any research on the central nervous system invasion that this virus might be capable of? It seems to be very adept in infecting many cell lines with the particular receptor, which is called ACE2, and wondering if there's any treatments or interventions in process or study that can address that. My understanding is that there's not really work on ACE2 receptor blockers that's far enough along to where we're ready to treat people with those. I will say research here at the MMRI, I'm part of a project that'll hopefully be out later this month that actually profiled myriad organ systems, including the heart, examining expression of not only the receptor, but other things that are very important to do kind of a pre-processing of the virus for infection. So there are a couple particular cells within central nervous system that are capable of uptaking the virus, but we'll have to go through a lot of the same effort to prove that it's actually active infection of that as well.
Okay, thank you so much, Nate. Our next question was if this was similar to old people getting pneumonia when they're lying in bed. And I think I'm happy to answer that question. Well, all pneumonia is basically caused by germs or bacteria and viruses. And what happens is people age is that they are more frail and they don't have the ability to expel those viruses or those bacteria from their lung as easily as younger people. And so when they're laying in bed, they aren't as active and they're no longer expelling as well. And so therefore the bacteria end up caught up in the lungs a little more frequently, and the viruses more frequently than those who are more active. This is also a consequence of aging, and which is why, as your question alludes to, why COVID may be more impactful and hurt more people who are older than those who are younger. The more active you are, the more likely you are to have the capacity to expel those things from your chest. But we're learning as we go, and I don't know if my colleagues want to address any more to that question. Okay. The next question, I'm going to focus on Chase. Why do statins not lower heart attack deaths?
That's a difficult question, but the real answer is that statins are there to help, as everyone knows, originally to lower your cholesterol, which then would help reduce your risk to create basically these injured areas in your coronary arteries that basically lead to heart attacks. So with that being said, there's a bunch of other things that we won't get into that contribute to heart attacks and heart attack deaths other than just reducing your cholesterol. I did mention that they also have effects on inflammation and things like that. So a person on statins actually does have a reduced risk. I think it's almost 50% if you're in the lower digits for cardiovascular disease and you're on a statin as well. So it does help a lot. That being said, you know, everything we say here is from, I'm a PhD and not an MD. So yeah, that would be the main reason why it doesn't actually lower heart attack deaths directly, I guess you could say.
Thank you, Chase. The next question is to Sathya. How does inflammation actually work to protect the body?
Oh, that's a great question. If you think about it, I call inflammation as a double-edged sword, because on one hand, it was originally designed to protect our body, but when it goes out of hand, that's when bad things happen. So how does it protect the body? Let's say there is an infection, there is a pathogen that entered your body. You need a system that can immediately respond and clear the pathogen off. So this is where your dendritic, I'm going to get a bit into science, so please bear with me here. This is where cells like dendritic cells or macrophages, they just go in and they engulf that pathogen and they digest it with acids. So that is how inflammation was originally designed to protect the body. But in this case of COVID, what happens is that there is so much infection that there is a huge, almost like a deluge of these inflammation cells, and the inflammation goes out of hand, and that's when it starts damaging the body.
Exactly. So thank you, Satya. It's called a cytokine storm. I don't know if you can touch a little bit on that with respect to why the inflammatory response becomes deleterious.
Sure. So all these cells have a mechanism to communicate with each other. So these so-called protective cells, these are like soldiers of a body. They're spread all over the body. And whenever someone senses a danger or a foreign pathogen in one region of the body, they need to communicate to the others to come to that region. And the way they communicate is through chemical signals called cytokines. Now, when you have a virus, and the virus, let's say SARS-CoV-2, is in the lungs and it's replicating massively, you have this SOS signals going from the lungs. So you have all these immune cells going into the lungs trying to protect by killing the virus. But what's happening in the meantime is this communication signal, what we call cytokines, they are secreted in such large amounts that the body can't handle it anymore. And that's why they call it a cytokine storm. And that leads to this uncontrolled chain reaction of damage that occurs to our body by inflammation.
Thank you, Satya. It's a good explanation. The next question says, I'm wondering about the fibrotic effect of COVID in the lungs, and can fibrotic lung tissue transformation be reversed? And I'll start that answering that question because I think it's a very good one. And the answer really is the jury's still out. There's active research currently being investigated on a number of tissue reversals of fibrosis and potential therapeutics that can be used to reverse this effect. Fibrosis is another word for scar. Basically, it's a way for the structure of the tissue to hold itself together in the onslaught of a stress or an injury of some sort. And so it's really, if you think about your skin scarring, some individuals have scars on their skin that may heal more robustly than others. Others have scar formation that continues to grow. I think that's very similar to what happens in tissues. And whether or not with respect to COVID, we can actually see a reversal of the tissue damage that is caused in the lung as a consequence of the infection, I think remains to be seen. It's an active area of research for Chase, I know. So I don't know if Chase, you want to answer any more to that question.
Sure. As you could imagine, the body is very good at healing itself. And the first step to that is creating these fibrotic lesions. The body then goes ahead and heals them away, usually by eating them back and putting better tissue. In the case of the lung, what we've seen via SARS 2003, the first one, is that the lungs can heal, but in some individuals, it's still impairing their function. And basically, that is due to fibrosis that has stayed due to the infection. The idea of my research and its aim is to say, okay, those that are healing from the illness, those that are recover, can we give them something at the time of their infection to lessen the fibrosis while also lessening inflammation damage, but not causing such an inflammatory reduction to where we hinder their ability to fight the virus? So there are these small studies using statins, but there's also other anti-inflammatory drugs that have been tested on other illnesses that have these kind of beneficial effects to where they don't knock it down completely, that is the inflammation, they just knock it down a little bit. So that's where science, we're really digging in to try to find the perfect balance for what that person needs when they come in. And with time is the only thing that we can say, because let's see, that was 2003 and the last study was just published right before COVID-2 came out. So that was 15 years and we're just now getting that answer, so.
Yeah, thank you, Chase. I think that dovetails pretty nicely into the next question, which is, if someone's been on statins for years, is there any data that will suggest that they will have a less effect with COVID-19 infection?
Right, so that would be something to where you would be able to data mine all of the data that's collected in, say, studies like the Framingham Heart Study and things that like Dr. Tucker, Nathan has been able to acquire. So those are just questions we all have in our minds, or at least I do. And then, you know, we just have to link up with the correct investigators, like ones here at the MMRI, to answer those questions by just digging in and looking at all the data and trying to make or see those relationships if they exist.
Yeah, thank you. I'm going to switch over real quick to a question that came up in the chat room about whether or not somebody should continue getting their annual flu shot. And I think that answer that we can probably all agree that more than ever, this is the best time to make sure you get your flu shot. I think the conjunction of flu and COVID can be devastating in the next few months. And the more we can protect people from getting the flu, the more we can tackle the number of people who are going to be infected with COVID as the cold temperatures also come back. And we're more likely to be indoors and touching and having the heat kind of circulate a lot of those particles, I think, more readily than being outside. Nate, I don't know if you want to answer any more to that question about the flu shot.
Yeah, I can't suggest it enough, especially in this time. The other, you know, from a testing standpoint and the hospital standpoint, things are really hard. So anything we can all do together to help reduce the probability that we're dealing with flu in addition to coronavirus, you know, I'd encourage everybody, you know, in any given year to get the influenza shot, but especially in this year, please.
Yeah, thank you. And the next question is also for you. Have you found any difference in viral RNA detection between nasopharyngeal or oropharyngeal samples? And do you find one to be better than the other?
Yeah, so there's a few different ways that you'll get tested. The vast majority will be taking essentially a Q-tip and sticking it in your nose until basically they feel something at the back of your nose, swirling it around uncomfortably for a couple seconds, and then repeating it in your other nostril. We have found effectiveness in other types of samples, including oropharyngeal swabs, including sputum samples, which is just spit. But you know, the amount of virus in each one of these is different, and I have seen the best data on the nasopharyngeal swabs, and I really hope that we continue using them. You know, when as difficult as everything's been, now is not the time to switch up the protocol we use for detecting virus, and that is certainly the place that we're getting the best detection right now.
Thank you. And the next question is similarly with respect to ACE2 receptor. Robert Harris asked, he suggested that he saw at Johns Hopkins that endocytosis occurs where the virus starts to replicate RNA. So with any antiviral, will that actually have other consequences due to that viral replication process? Or is blocking the ACE2 receptor the best approach?
So I can tackle a little, yeah. The coronaviruses are special, being RNA viruses, as you obviously know, Mr. Harris. So blocking the receptor at this time doesn't seem to be at least an option for this particular outbreak. Working on viral replication can have, you know, what it's basically doing is hijacking some of the machinery that's normally in the cell. And when you start to block these things, there's what we call off-target effects, and in reality and practice turn into the long, fastly read disclaimers at the end of pharmaceutical commercials. So, you know, the vast majority of people are you know, don't have a lot of symptoms and are do well with the virus. I think, you know, where we start dealing with some of these other more aggressive treatments or with people who are having a more severe episode. So it's a risk benefit with anything, you know, when you're examining a treatment. I'll address the second part of the question too. Once the virus is in the wild, isn't it true that we'll all have it? That's the one outcome that is not the best outcome. So that's a thing we call herd immunity. And depending on the virus, it needs to reach a certain number of people before we basically get a certain amount of protection. So it'd be similar to the herd immunity that we've established in recent times against things that used to be terrible for humans, like polio, or other childhood illnesses that we've now mostly eradicated with vaccines. The hope is that we're able to deliver vaccines before we reach that herd immunity point, especially when we're seeing death rates from this up to at least in New York State, 0.6% of people who are contracting the virus are actually dying of it. So that is certainly a possibility, although not the best outcome, I think.
Yeah, I completely agree with that sentiment. And in fact, most recently I heard the number is closer to 60 million people would have to be infected worldwide before we could reach herd immunity. And currently, despite the devastating effects of the virus, we're somewhere between 5 and 10 million people worldwide. So it's still not it's not enough to get to that point. We'll probably get to a vaccine sooner. And that's the hope, I think, with all of the research that's currently ongoing. The next question is from John Shikita. For that study that came out 2 weeks ago about the 100 patients with heart issues at 2 months post-infection, there was something like 72 with heart issues. This seems really high. Was that just a random sampling, or is this going to be a chronic issue that many people with cardiac conditions will have to deal with?
Sure, I can take that. That's a great question. But like you said, the sample size is 100, and in science, that's too small a sample size to draw any meaningful conclusions. I would say it's suggestive. If I have to speculate, I would say, especially in people who had severe infections, who were in the ICUs, who were on ventilators, they might have some cardiac issues down the line, but that is something I think doctors are looking at closely and are monitoring based on studies like these, whether they have to do a long-term monitoring of the heart. It's too soon to conclude anything based on one small study.
Thank you.
I'd like to make a little follow-up to that one too, if you don't mind, Maria.
Sure.
Please.
One thing I'd like to say to everybody watching too, that there's a certain rush to sensationalism with not only a lot of the media coverage of the virus spread itself, but also for a lot of the research done about it as well. So when you see small studies like this, to, I guess, temper how much you're crediting one individual report from one individual study until science needs, this is a thing we're poorly adapted to, but we have to come to a consensus about what's happening. And just now, some of the earliest things we understood about the virus, we've actually come to a consensus on. And, when we're talking about these heart issues, a study with 100 patients, I'm glad I didn't review it because we probably wouldn't be reading it right now. But it's a bit of sensationalist stuff that's coming out right now. So I'd say to everyone, it's hard times and everybody's excited, but be as patient as you can until we come to the actual conclusions.
That's a good response. The next question has to do with COVID-19 research on those affected with diabetes. And maybe some of my colleagues will answer, but I'll start. We're currently, as far as I know, not working on diabetes specifically as it relates to COVID-19, but there is a lot of active literature and research currently being conducted on the role of diabetes in patients that have COVID-19. And the understanding is that it is a pre-existing condition whereby those that get infected with COVID-19 have a worsened outcome. So it is an important characteristic to be looking for. Diabetes, hypertension, cardiovascular disease, and other ailments, obesity and diabetes being, again, one of the major ones, has been associated with increased propensity for those that end up in the hospital with COVID-19. However, above diabetes, I will tell you those that have hypertension are more likely to end up in the hospital than those with diabetes alone. But I don't know if Nate, Sathya, Chase, you have anything you want to add to that?
Nothing from my side. You covered it pretty well.
Okay, good. Next question from Roger Frank. Can long-term damage be repaired with stem cells? Very interesting question. Does anybody want to take that on? I can take it because we're doing a lot of active stem cell research currently. Again, like the fibrosis question with respect to reversing scar tissue, I think that this is we will find out, and it will take time for us to figure that out. As we're starting to do a lot of stem cell therapy, we're beginning to realize that stem cell therapy does not have a lot of efficacy in the heart. So you can't really reverse heart disease using stem cell therapy, although in this case, when you inject a heart with stem cells, the injection alone has been reported to actually have a positive effect because I think, as one of the earlier questions suggested, inflammation, at least a small amount of inflammation, can be protective. And it turns out that that inflammatory response can actually help reverse some of the effects of heart disease. Similarly, that may be true for lung damage as a consequence of COVID-19. But as Sathya mentioned earlier, that will depend on the intensity of that inflammatory response and whether or not we want to be able to go back in and induce an additional response that may have more deleterious effects downstream. So I don't think we know the answer to that fully until we do the experiments. Next question, no mention of diabetes, since this is mentioned with underlying heart condition as two conditions concerning elderly and COVID. I think we addressed part of this before. We have a couple of people here working on diabetes and obesity. I think your questions may prompt them to want to consider looking at it in the context of COVID-19. But again, it is a very active area of research whereby we do know there's an association and likelihood that those patients with diabetes will have more severe COVID-19. Any vaccine available soon? Nate.
Yeah, so the Moderna one that Maria alluded to earlier, so that's an entirely new form of vaccine that actually uses RNA to try to stimulate a response. So there's never been a vaccine like that before that's made it to market. That entered phase 3, which is the last phase before you get a wide release to people. My understanding is they hope to start getting a lot of that data back sometime in the middle of this fall. My understanding, I believe Pfizer has another vaccine that's in phase 3 as well. So there's certainly, from a shots on goal standpoint, a lot of investment and a lot of work going into vaccines. You know, hopefully we get ones that work is the most important thing. There'd be nothing worse than a vaccine that we give to 100 million people that does absolutely nothing. But if I think, you know, between the ingenuity and investment in all these companies, hopefully, you know, later on this year and into early next year, some of us will, especially the high risk people, will be receiving a vaccine.
Thank you. And I think the last question is for you, since you brought up COVID-3. When do you think that might be a problem?
Yeah, it's a dice roll. These things are like earthquakes, right? So the California earthquake that came early this morning, you don't know when they're going to happen, and there's a probability they can happen at any moment. There's a chance that SARS-CoV-3 comes next year, and there's a chance that SARS-CoV-3 doesn't happen until I'm dead and gone. So really all we can do is learn what we can now and prepare as best we can so that all these infrastructure problems and all the misunderstandings of biology that we had going into this thing are done by the time that SARS-CoV-3 comes around. So that's, we will operate as if it's coming next week, and hopefully it doesn't.
Thank you, Nate. There's one more thing I want to add to that in a conversation I had with Grandmaster this morning. First of all, we're subjected to viruses and bacteria, new viruses and bacteria every day. We are built machines that have the capacity to fight the majority of these things. It's not every virus or every bacteria that we need to be worried about. On occasion, there are things that our immune systems just haven't seen before and respond in a way that we can't possibly predict, such as these SARS viruses that have come along, and all of them have been pretty severe from the first to the current SARS viruses. But as Grandmaster said this morning, one of the things that is one of the reasons why our pandemics may be coming sooner rather than later is because the world has gotten smaller. We travel like it's an afterthought, or we did before this epidemic. And so we will get on a plane and cross the country and go over to another continent in the same day. So because of that, the transmission rates for these things will be a lot quicker than we were used to for the Spanish flu, for example, in 1918. So these are things that I think we need to keep in mind. And as we continue to resolve a lot of these diseases over time, we'll figure out better ways to combat them and hopefully, as Nate said, plan for them as best we can in the future. So turning it back to you, Grandmaster, that was our last question.
Oh, I
Thank you
see a
very
few
much.
more.
Sorry, some came in.
Yeah.
Do children's immune systems react differently to the virus than adults? Jace.
Unknown.
I will say that they seem to be less symptomatic on the whole, and certainly have the incidence of mortality in the young is vanishingly low. So something certainly different between the response of the young and the old.
Yes, but I do want to say with caution that just because their immune system is protective doesn't mean they aren't carriers. So I think that one of the biggest concerns that everybody has on their mind these days is about school reopenings and what that might mean. And yes, it's true that children are less likely to have severe effects of COVID-19, but they can bring it home to elderly, and they can bring it home to people who are susceptible and who have vulnerability to the virus. So it just because they themselves may not have a very severe outcome doesn't mean that they may not be able to pass it along to someone else who might, which is one of the major reasons why we're really pushing for people to be wearing masks, because it's not about protecting yourself, it's about protecting those around you, because you just don't know who is susceptible to this virus. And you just can't predict that these days, at least not yet. Next question is for Sathya. How can you explain the difference between T cell and B cell immunity?
Wow, it's almost like a one semester course in a university.
Get ready, everybody.
No, so I'll try my best to keep it brief and still keep it meaningful. So in our body, we have two types of immune systems, broadly classifying the innate and the adaptive. The innate is a dirty bomb. The adaptive is like a laser guided missile. Like it's very targeted. That's why it's called an adaptive immunity. Now in the adaptive, you can again classify it into two classes, the T cell and the B cell. Depending on the region of the body where they mature, the T cell matures, those cells mature in thymus, and the B cells happen in a bone marrow. So in T cells, again, there are two classes, the CD4 T cells and the CD8. The CD8 is called cytotoxic cells. These are the cells that actually go and eat up the pathogen or kill the pathogen. Now in the B cells, when you talk about antibodies, you're talking about the B cell immunity. That's where the antibodies, that branch of immunity is what generates the antibodies. But other than that, I don't know what else can I add. Do you want to add anything else, Maria?
I think that I learned a lot. I think that's terrific. That was a really good explanation. And
I think the only thing to add is that the adaptive immune system takes time for your body to recognize the pathogen, process the pathogen, and then form some kind of antibody against the pathogen. So that's kind of that lag period that we're seeing where people either get better at these teens days after infection, or people progress to worse because their adaptive immune system has been either hindered or, is working well. So.
And maybe
I think we'll take one more question, and I think people have other things that they got to go back to tonight. But I think we have one more from Thomas, and then we can close out with some good comments.
perfect. So the last question, are there any particular blood types susceptible to COVID-19? So yes, but recently maybe that has been contested. So the answer is yes, type A blood seems to be most susceptible to COVID-19. Type O blood seems to be the most protected. But recently there was a publication that suggested that maybe the vulnerability to type A blood with COVID-19 may not be as high as anticipated before. I don't know if Nate, you were nodding. I don't know if you want to add anything more to that.
Yeah, it's a good, I mean, that initial study was certainly, I think that speaks to the timing of this too. I think that was 2 weeks ago. But that original study was linking type A or type AB blood to an increased risk happened. And, you know, again, it's a good point to say, let's relax and wait for better data to come out in order to jump to these conclusions. But
so this has been great. I just want to thank my faculty for taking the time this evening. Grandmaster, thank you for having us as part of your wonderful series. And we're grateful to you and to this opportunity, and turning it back to you.
Well, thank you very much. I'm going to end here with, usually when I start talking now, people start logging off. But there is something here. Alden says, I would just want to thank you for each of you. You are heroes behind heroes. If not for research and translational science, we would not be able to have evidence-based interventions in clinical practice. Well, Alden and Dr. Maria are two people I will not play Scrabble with, that's for sure. I do enjoy when I listen to Dr. Maria talk about your backgrounds, because you can put my background on the back of a matchbook cover. So your backgrounds and what you've gone through are terrific. I think there was a slide that was up there. What you're doing doesn't come along cheap. What comes along is it needs money. And I think there was a slide that had a MMRI EDU giving slide. I think that was up there. And what you can do is research is what they're doing. And at the end of the research, there's what we have is the light at the end of the tunnel. For those of us who are out here in the field, we look to you and others as the ones that are going to save humanity. Let's be honest. And Masonry can save humanity on one end of it through the bond of brotherhood and helping each other. But the science of what you all have together and what you employ and deploy is really what makes a difference. I know that you've all been taking time during the day today to practice this. Make sure your hair was combed. Make sure you had the right background. Make sure your headsets were good. And you took a lot of time out of your busy schedules. What should be noted is that the MMRI is doing more than just this. There are many balls in the air that they are doing research on in this institute that we're not going to go into tonight, but we could make another night for this at some point. And I want to thank all of you. I know you all have places to go tonight. I know you all have families. I know you have things to do. I appreciate it so much that you're here. God bless each and every one of you for what you do. God bless our great Masonic fraternity. And God bless the United States of America. I appreciate your time and patience, skill, and passion. Thank you so much.
Thank you.
Thank you.
thank you.